Hello, and welcome back to Introduction to Genetics and Evolution. In most of the videos that you've seen so far, we were studying patterns of genetic inheritance, looking at alleles from one gene at a time. In the last video you saw, we studied patterns of genetic inheritance, looking at two genes at a time rather than one, but focusing on genes that are on different chromosomes. And we discussed how those assort independently. They follow Mendel's law of independent assortment. In this video, we'll continue to look at patterns of inheritance of two genes at a time, but looking at genes that may actually happen to be on the same chromosome, but maybe not right next to each other. Now, this is called crossing over, and this is a subset of the overall process of recombination. Now, many of you use Wikipedia, I know. Recombination, you can basically break up the word as I've done here. Re for a new, combination. So basically recombination is shuffling of alleles into new combinations, combinations that were not found in the parents. I will emphasize the Wikipedia definition in this regard is not precise. The Wikipedia definition focuses specifically on one type of recombination called crossing over, which will be our primary focus. But I want to emphasize that all recombination is important, both for genetics and evolution, and independent assortment is one type of recombination. Essentially, you have recombination when you produce a gamete that has a combination of alleles that was not found in one of the parents. This gamete is then called recombinant. Okay? So you have to examine this by looking at essentially two stages. You have to look at the person, the sort of focal person. You have to look at the gametes that made this person, things from their mom and their dad, and you have to look at what happens in their gametes to see this. So let me illustrate this with an example. Let's say that you're looking at something on the X chromosome, which is one chromosome, and let's say you're looking at the two traits that I talked about in the last video, being colorblind versus not colorblind. And let's say you're also looking at a trait that's controlled by one gene, and something similar to what I described before with the left thumb landing on top, as opposed to the right thumb landing on top. Remember, the one with the left thumb landing on top was dominant. So if you have a sperm from somebody who is not colorblind and with left thumb on top, so this is the sperm that's coming in. Here's the egg with the X that's colorblind and right thumb on top. You end up with somebody who's heterozygous for both, right? He has one copy of each allele. Now, in this case, this individual who's a female is going to have an egg. Note now this egg will through this process of independent assortment is just as likely to give this X as that X. In this particular case, this X is the one that lands in the egg, and this other chromosome that has the left thumb on top is also the one that lands in the egg. Note this combination is found in this egg, the XCB and the L. The XCB actually came from the mom, from this individual's mom. The L came from this individual's dad. So we actually have a new combination. This is a combination that was not one of the forms that made the person, right? This was not something found in the parental sperm or egg, but it is a new combination. That is why this is called recombinant, right? Because we do not see anybody up here who's XCB and capital L. So what's the opposite of a recombinant? The opposite of recombinant is a parental type or non-recombinant. So the first part of this figure is exactly the same as the previous one I showed you. But in this case, what happened is this ended up in the egg and this ended up in the egg. Now these two are the exact combination that was found in one of the parents, right? So this would be considered a parental or non-recombinant gamete. Do you see what I mean? So basically what you're looking at the gametes that made the individual and the gametes that are coming out of the individual, and you're looking to see, is there a new combination in the gametes coming out of the individual? If there is, then you've had recombination. Now there's two means of recombination. One of them is independent assortment, which we've talked about, and this is typically what happens when you have different chromosomes, right? That you're just as likely to pass on one copy of the chromosome as another copy of the chromosome based on Mendel's laws. Now, the other possibility is crossing over. Now, again, sometimes the word recombination is synonymized to this. Sometimes people, when they say recombination, are referring specifically to crossing over. In this case, you're actually breaking off a piece of one chromosome and sticking it to another. This right here is a picture from Thomas Hunt Morgan's book from about 1916, where there's a white chromosome and a black chromosome. Pretend that one came from the dad, one came from the mom. And you see in the offspring, you may get this part black, part white chromosome. So it's this, again, new combination, but it's actually it's happening along the length of one particular chromosome. So let me recap what these two things are. First, in terms of independent assortment. Importantly, we can study independent assortment by multiplying probabilities, right? Because we assume that traits are inherited independently. So again, if you're just as likely to pass this gamete or pass on this allele as that allele, we can then just multiply the probabilities to get what's going to happen among all the offspring. So in this example here, this is one of the ones we did in a previous video. If you're heterozygous for both the T and S, this is what would happen in terms of T's. This is what would happen in terms of S's, right? If you're big T, little t, cross to big T, little t, 1/4 of your kids will be big T, big T. 1/2 will be big T, little t. 1/4 will be little t, little t. Same thing is happening with S's. So then to see what would happen for the combinations, we just multiply the two things together. So your probability of being big T, big S would be 1/4 times 1/4, or 1/16. That's what this is right here. All of these are divided by 16. So 1 would be this. Your probability of being little t, big S would again be 1/16. Again, right there. Your probability of being big T, big S, little s, that's 1/2 times 1/2. So that would be 1/4, or 4/16. That's what we see right there. So this assumes true independence. Now, independence works well with this independent assortment thing. And we can see how this happens by looking at the chromosomes. Imagine here, let's say all the blues came from dad and all the reds came from mom. And this is now in the individual. This is chromosomes 1, 2, 3, and 4. Okay? You have your meiotic divisions, and then you produce this set of gametes. You can see along here, you may pass on the red chromosome 1, you may pass on the blue chromosome 3, you may pass on the red chromosome 4, etc. All sorts of different combinations happen. But in reality, when you're looking at genes along a chromosome, it's not that simple. Often homologous chromosomes, these chromosomes that basically that came from dad and came from mom, will trade pieces during this process of meiosis. And that was what was depicted in that diagram from Morgan. So this happens when they're doubled, and this process is called crossing over. And this again forms recombinant chromosomes, or recombinant gametes, where if you're looking at two genes, you can get combinations not found in the parents among the alleles. So let me just walk through this a little bit more slowly. So you have two homologous chromosomes, one inherited from the egg, one inherited from the sperm. So here's your egg chromosome, here's your sperm chromosome. We see them both. So here they are, they're put together. Okay. So what happens then in the individual, so let's say, for example, this is me, and the red I got from my mom, the blue I got from my dad. What may happen inside me in the process of making some of my gametes, I may have something like this happen, where pieces get broken and stuck together, such that among my eggs, I obviously didn't produce eggs, but among my eggs, I might have some non-recombinant, so this is something that passing on exactly what I got from mom, and some that are recombinant, which have bits from mom, bits from dad, bits from mom, bits from dad. This is a recombination event happening along a chromosome. So you may be wondering, how do we tell if recombination has happened? How can we tell that this is going on? Well, what happens is we look at genetic markers. And we'll talk about what genetic markers are, but you can think of them as sort of the big A, little a, and big B, little b that we've talked about in the past. You could think of them like the traits they're associated with, which thumb's on top, or being colorblind or not. And in this case, let's say, for example, we know that from your mom, you got the capital A and capital B. We know from your dad, you got the little a and little b, right? The non-recombinant eggs from this individual would be capital A, capital B, and little a, little b. Again, something that was identical to the sperm or egg that made you. The recombinant eggs would be those new combinations, little a, big B, and big A, little b. Okay? Now, importantly, you won't always have recombination. In fact, if let's say, for example, the A and B genes were right next to each other, let's say they're basically stuck together, at that point, you refer to them as being totally linked. They're linked together, that if you get the big A, you're always going to get the big B. If you get the little a, you'll always get the little b. If that was true, then you would only get these non-recombinant eggs, right? If basically these two things were glued together, then you would only get non-recombinant eggs. And what tends to happen is neighboring gene variants, or alleles, tend to stay associated. So I've diagrammed here what you might imagine looking at several chromosomes from one heterozygous individual. So again, here's the chromosome from mom on top, that's red. The bottom chromosome is from dad, that's blue on the bottom. They are producing gametes. You might see this along the gametes there. So you have a stretch of red, stretch of blue, stretch of red, stretch of blue, stretch of blue, stretch of red, etc., etc. If you look at this, how often are A and B linked? How often do you inherit a red A with a red B, or a blue A with a blue B? Now, A and B are right next to each other, and if you look across this whole chart, pretty much always. If you get a red A, you're always getting a red B. If you're getting a blue A, you're always getting a blue B. You see that very clearly along here. In contrast, how often are A and C linked? Notice A and C are further apart. Well, here in this first individual, A and C are both red. In the second, we actually get a red with a blue. Third, we get a blue with a red. Fourth, we get a blue with a blue. So really only about half the time are you getting the two parental types, and half the time you're getting a recombinant type. Now, this is what's used for constructing genetic maps. That if you have genes close together, they tend to be inherited together. You're violating this principle of independent assortment that I described before. Though what's happening in this case is you're looking along one chromosome rather than looking at different chromosomes. But you can no longer multiply the probabilities like we did before. So let's try to follow this. Now let's assume total linkage. Let's assume that A and B are stuck together. They're right next to each other. So let's imagine dad is big A, big B. Let's imagine mom is big A, big B, little b. So dad is always giving the same gametes no matter what. Dad is always giving a big A, big B, right? So we see that along this chromosome. Dad is always giving a big A, big B. Mom, in this case, will only give a big A, little b, right? Or a little a, big B, because we said it's linked, right? So this is what mom's going to give. So what kind of offspring can we see? Well, we can have big A, big B, little b, which is the same over here. Or we can have big A, little a, big B, big B. So in this case, there's only two possible forms that you can get out of this. There's only two possible offspring if you have total linkage in this case, right? What would happen if, in contrast, there was no linkage? What would happen then? Well, again, dad is always going to be giving big A, big B, right? Because that's all he can ever give. Even if he did have a recombination event, we would never know because it would always still be big A, big B. But in this case, mom has more things she can give. Here we have the parental types, big A, little b, and little a, big B, right? But if there was no linkage, sometimes little a and little b can go together, or big A and big B can go together. So these are recombinants, and these are parentals. And as a result, there's a lot more possible options that you can get out. So we can get, in this case, big A, big B, little b, big A, little a, big B, big B, big A, big B, big B, and big A, little a, big B, little b. So in this case, now we have 4 possible types of offspring rather than just 2. Now, one thing I hope you noticed in looking at this is that phase was important, that basically which allele is next to the other one. In this case, let's say, for example, you were to say an individual is big A, big B, little b. You don't actually know the phase. You don't know what combination of alleles he got from the sperm that made him as opposed to from the egg that made him. In this top example, the egg that made him was big A, big B, and the sperm that made him was little a, little b. So that's one possibility. The other possibility is the egg that made him was big A, little b, and the sperm that made him was little a, big B. In each of these cases, you get different sorts of recombinant gametes. These bottom ones, let's look at the bottom set. The capital letter ones are recombinant gametes here, right? Because you have to have a recombination event to get those. For the top one, the one capital with one lowercase is what's recombinant. So it's very important that just because you see an individual as big A, big B, little b, you can't actually just from that alone figure out what sort of recombination is going on. So we'll pick up on this in the next video to look at how to calculate recombination distances between genes. Thank you very much.